All right, let's jump into another deep dive. This time we're tackling evolution, specifically evolution and phylogeny.
Always a fascinating topic.
Absolutely. We've got some excerpts from a college-level evolutionary biology lecture. Dense stuff, but really intriguing. And what we're going to do is try to, unpack it all, trace those branches of the tree of life.
That's a speak.
Speak.
Yeah.
We'll see how scientists actually put together, figure out evolutionary history, learn how to read those family tree diagrams.
The phylogenetic trees.
Yeah, phylogenetic trees. And get this, we might even uncover some surprising evolutionary relationships that'll make you question what you thought you knew about, the connections between different creatures.
You'd be surprised how often looks can be deceiving in the animal kingdom.
Right. So, okay, let's start with the basics. Like, if we're going to understand how species are related, first we need to be able to identify them, right?
Of course.
Like, what even makes a robin a robin?
Well, that's where things can get a bit tricky. Common names, they can be really misleading. I mean, would you believe that a robin in North America is a completely different bird from a robin in Europe?
Really?
Yeah, to get around that confusion, that's where scientists use, binomial nomenclature, that genus-species thing.
Right, like Homo sapiens for us.
Exactly. It's like a universal language. Make sure everyone's talking about the same organism, no matter where they are.
It's like a unique ID for every species, basically.
You could say that.
So we've got these names, and of course that's part of a bigger system, right? A classification system.
Oh, absolutely. Going all the way back to Linnaeus and his system of classifying life.
Right, with all those different levels like kingdom, phylum, class. It can be hard to keep them all straight.
It's like those Russian nesting dolls. Each level fitting inside the next, encompassing a broader group of organisms.
Right, right. So it seems pretty straightforward. We've got these neat little categories for everything, but I'm guessing it's not as simple as just sorting organisms into boxes based on how they look, is it?
You're catching on. While the Linnaean system is helpful for organizing things, making sense of the diversity of life, it doesn't always line up perfectly with evolutionary relationships.
So how do we figure those relationships out?
Well, that's where things get really interesting. We need a system that digs deeper, something that can show us those hidden connections between species.
Enter the phylogenetic tree, the family tree of life. I love that visual.
It's a powerful tool.
Instead of those awkward family reunions, we've got this incredible diagram showing how everything is related, right?
Exactly. It maps out those evolutionary relationships, showing us how species are connected through their common ancestors. Scientists use a mix of things to build these trees.
Like what?
Well, there's morphological data, basically physical traits, what the organism looks like.
Right.
But they also use molecular data these days, like DNA and protein sequences, comparing those between different species.
So it's like detectives piecing together clues from both the outside and the inside.
That's a great way to put it. And one of the most interesting clues they look for are these things called homologous structures.
Homologous structures.
Similarities in structure, like bone arrangements, that point to a shared ancestry, even if the function's totally different.
Like, even though a bat's wing, a whale's flipper, and a human hand look and work so differently, they're all built on a similar bone structure.
Exactly. A blueprint passed down from a common ancestor, modified over time.
It's like inheriting your grandma's famous cookie recipe, but you tweak it to create your own signature twist.
I like that. But you have to be careful. As you get deeper into these relationships, sometimes looks can be deceiving.
Oh, you're talking about those glass lizards.
What about them?
The ones that look just like snakes but are actually more closely related to, wait for it, lizards. It's like nature's playing a cosmic joke on us.
Well, it all comes down to convergent evolution. You see, similar environments can push totally unrelated organisms to evolve similar traits. Like that long, legless body plan works great for slithering through grass, whether you're a snake or a lizard.
So how do we tell the difference then between those truly inherited traits and the ones that just happen to look similar because of the environment?
That's where the real detective work comes in, separating those inherited family heirlooms from the copycats.
And that's a story for part 2, I guess. We'll continue our deep dive into evolution and phylogeny next time. All right, so we're back diving deeper into evolution and phylogeny. Last time we started to untangle the tree of life, saw how scientists use those phylogenetic trees to map out evolutionary relationships.
We even touched on some of those tricky cases where appearances can be deceiving.
Right, like those glass lizards. They look like snakes, but they're not. So we left off with this idea of cladistics, this method where scientists look for shared, newly evolved traits, like those unique family heirlooms, to, define specific branches of the tree.
It's all about finding those key characteristics that set one group apart from another.
But how do you even begin to sift through all those potential traits? I mean, how do you decide which ones are actually useful for building these trees?
It's a bit like trying to trace your own family tree, right? You wouldn't use something like having two eyes or walking on two legs to define a specific branch of your family.
No way, those are way too common.
Exactly. You'd be looking for those unique quirks, the things that make your family, well, your family, like your great aunt's obsession with collecting porcelain frogs, or maybe your uncle's uncanny ability to wiggle his ears.
Okay, I see what you mean. So in the world of phylogeny, it's all about finding those quirky traits, right? The ones that really tell a story.
Precisely. Those are what we call shared derived characters, traits that evolved in a common ancestor and are shared by its descendants. They're the gold nuggets that help us define specific groups on the tree.
But wait, we were talking about how not all similarities are created equal, like those celebrity lookalikes. They might resemble each other, but they're not actually related.
Right. And the same thing can happen in evolution. It's called homoplasy.
Homoplasy.
Yeah, traits that look similar but actually evolved independently. Like with the glass lizard, that's convergent evolution. But sometimes it can happen through evolutionary reversals where a lineage loses a trait that was present in its ancestors and then later on it reappears.
This is like nature's playing a game of copycat trying to trick us.
You could say that, and it makes figuring out true relationships really tricky. Distinguishing between homology, true shared ancestry, and homoplasy, that's super important for accurate tree building.
So we can't just look for any old similarity. We have to figure out if it's a meaningful similarity.
Exactly. We have to determine if it points to short ancestry or if it's just a coincidence. And to do that, scientists have a few methods up their sleeves. One of the most common is called maximum parsimony.
Maximum parsimony. That makes sense.
It's actually a pretty simple idea. It's all about finding the simplest explanation. In phylogeny, that means the tree with the fewest evolutionary changes.
So like the simplest evolutionary path, the one that makes the most sense.
Exactly. It's like finding the shortest route on a map. You want to minimize the number of steps, or in this case, evolutionary changes, needed to get from the common ancestor to the present-day species.
Okay, that makes sense. But I imagine things can get pretty complicated when you're dealing with tons of species and all their different traits.
Oh, absolutely. Luckily, these days we have powerful computer programs to help us crunch the numbers. They compare different possible trees and see which one requires the fewest evolutionary changes.
It's like having a team of superpowered evolutionary detectives on the case. I remember the source material mentioned another method called maximum likelihood. Is that kind of similar?
It's related, but it takes a slightly different approach. Maximum likelihood, it factors in the probability of certain evolutionary events happening. So some changes are more likely than others, based on what we know about genetics.
So it's like weighing the evidence, considering how likely different scenarios are.
Exactly. It's all about finding the tree that's most likely, given what we know about evolution. Scientists often use both maximum parsimony and maximum likelihood to see if they get similar results, kind of like getting a second opinion.
Two evolutionary doctors.
Yeah.
Better safe than sorry. Okay, so up until now, we've mostly talked about how to build these trees. But what can they actually tell us? Like, besides just who's related to whom, can we learn anything else?
Oh, absolutely. For one thing, we can use phylogenies to make predictions about evolutionary history. There's this classic example with dinosaurs, birds, and crocodiles.
Okay, this sounds interesting. What do those three have in common?
Well, based on a lot of evidence, both morphological and molecular, scientists think that birds are actually most closely related to crocodiles. It might seem surprising, but if you look closely, you can see some shared traits in their skeletons.
So are you saying birds are like fancy crocodiles?
Not exactly, but they do share a common ancestor. And that ancestor must have had certain traits that were passed down to both lineages. Now, dinosaurs are more closely related to birds and crocs than they are to other reptiles. So scientists predicted that dinosaurs might have shared some of those same traits, too.
So they were making predictions about dinosaurs based on their relationship to birds and crocs. Did they think dinos would suddenly sprout feathers?
Not quite, but they predicted that dinosaurs might have shared some of the reproductive behaviors of birds and crocs. Like maybe they built nests and cared for their eggs instead of just laying them and leaving.
Okay, so did they find any evidence to support this?
They did. There have been some amazing fossil discoveries that back up these predictions. For example, they found the remains of a dinosaur called Oviraptor sitting on a nest of eggs like it was brooding them, just like birds do today. And they found other fossilized dino nests with eggs carefully arranged. It really suggests that parental care was more common among dinosaurs than we used to think.
Wow, that's incredible. It's like those phylogenetic trees are not just maps of the past, but also like treasure maps leading to new discoveries.
They really are powerful tools. And we haven't even gotten to those fascinating molecular clocks yet, but I think that's a story best saved for the next part of our deep dive.
Okay, I'm already hooked. We've covered so much ground already, from those tricky copycat traits to dinosaur parenting. I can't wait to see what other evolutionary wonders await us in the final leg of our journey. All right, welcome back to our evolutionary adventure. We've journeyed through the twists and turns of phylogeny, learned how scientists decode those relationships, and even uncovered some surprises along the way.
It's been quite a ride.
It has. But today, we're zooming in even further, right into the heart of genetic innovation.
It's a fascinating world down there, a realm where genes are constantly evolving, changing, even duplicating themselves.
Yeah, and remember how we talked about those duplicate genes? They're like having spare parts, right? They can tinker with new functions without messing up the original gene.
Exactly. It's like evolution has this playground where it can experiment and try out new things.
And those experiments, well, they've had a huge impact on the diversity of life we see today, right?
Absolutely. Think about those globin genes, the ones responsible for oxygen transport. Hemoglobin, myoglobin, those are essential for life as we know it. And those genes, they got their start through duplications and modifications over millions of years.
So they basically evolved to specialize in different ways of moving oxygen around.
Exactly. It's evolution at its finest, constantly refining and tweaking its toolkit to adapt to different environments and lifestyles.
It's incredible how those tiny changes at the genetic level can lead to such big differences in an organism's form and function. Yeah. Speaking of tiny changes, didn't we talk about olfactory receptors? Those genes that allow us to smell.
Ah, yes, those. And in the world of olfactory genes, duplication has gone wild. Some organisms have hundreds, even thousands of copies of these genes.
So like mice, they can detect way more smells than we can, right?
Yeah. Oh, absolutely. Mice have over 1,000 functional copies of these genes, giving them a sense of smell that's orders of magnitude more sensitive than ours. We humans, we only have about 380 functional copies.
Maybe that's why I can never find my keys. But seriously, it's amazing to think about how gene duplication has fueled so much diversity. It's like nature is constantly shuffling its genetic deck of cards, creating new possibilities with every hand. But here's a question I've been wondering about. How do scientists actually keep track of all these genes? I mean, how do they know if a gene arose through duplication or if it was just inherited from a common ancestor?
That's a really important question, and it gets to the heart of how we trace the evolutionary history of genes. One key distinction is between orthologous and paralogous genes.
Orthologous and paralogous. Those are some serious science words.
I know, they sound a bit intimidating, but the concept is actually pretty straightforward. Imagine you have a gene, and it's present in the common ancestor of, say, humans and mice. As those lineages evolve, that ancestral gene gives rise to separate lineages in each species. Those are orthologous genes, like distant cousins who share a great-grandparent.
So orthologous genes are inherited vertically, passed down from generation to generation.
Exactly. Now, imagine that ancestral gene undergoes a duplication event in, let's say, the human lineage. That creates two copies within humans, and those are paralogous genes.
So like siblings, sharing the same parent gene.
Exactly. And distinguishing between those two types is crucial for understanding evolutionary relationships. Orthologs, they give us a window into how genes have changed since the last common ancestor, while paralogs show us how gene families have diversified and specialized within a lineage.
It's like having two different perspectives on the evolutionary story, right? This has been an incredible journey, gone from the basics of naming species to the mind-blowing world of genes and molecular clocks.
We've really delved into the interconnectedness of life, exploring the mechanisms that have shaped the incredible diversity of life on Earth.
It's a story that's still being written, right?
Right.
Every new discovery adds another piece to the puzzle.
Absolutely. The world of science is full of surprises, and there's always something new to learn, so keep that curiosity alive and never stop exploring.
Couldn't have said it better myself. That's a wrap for this deep dive into evolution and phylogeny. Thanks for joining us on this adventure.